And we have our final speaker of this session, Michael Ringel from our knowledge partners at the BCG. It's super exciting that you're here. You know, it's I think, a validation of our field in a way that a consulting firm like yours is interested in what we're doing. So please go ahead.
Yeah, thank you. So good afternoon, everyone, and it's really great to be here in person. It's an amazing group to be a part of. And to the folks that are joining remotely, hopefully next year you'll be in person. And big thank you to Morten, Daniela, and Alex for inviting me. So the organizers asked me to just share a bit of the context that sits behind, how the commercial market may evolve for therapies based on aging biology. So I thought I'd start by pulling back, way back, in fact. So this is the Earth forming 4.6 billion years ago. So hopefully you've got your cup of coffee and are settled in for what's going to be a bit of a journey here. How much time do I have, Morten?
We have dinner at some point, I guess.
Okay. So the great evolutionary biologist John Maynard Smith was among the first to point out that you can think of a series of major transitions in evolution, each of which were a great leap forward. So the first of these, shortly after the end of the late heavy bombardment, was the emergence of life. And not too long thereafter of the central dogma that DNA begets RNA begets proteins, likely from a precursor world where RNA did everything, right? The information storage of DNA and the catalysis of proteins. We then have the emergence of free-living life, possibly twice with the eubacteria and archaea. Complex cells with the symbiosis of mitochondria and chloroplasts and emergence of other organelles, multicellular organisms, and finally, societies. And what John Maynard Smith pointed out is that what each of these transitions shares, in addition to taking hundreds of millions or billions of years, is that they're all examples of cooperation coupled with specialization, coupled with the creation of a more effective whole. Whether that's RNA parsing out its duties to specialist DNA and proteins, whether it's specialization of organelles or cell types or jobs. So when we think about this context applied to the task in front of us of translating aging-based therapies into drugs, it at least is suggestive, along the lines of what Mahmoud pointed out yesterday, that there is an ecosystem that we need to build that has multiple parties, each doing their role. And in particular, that when we think about translation, how important pharma is going to be as part of the picture here. Incidentally, if you pull back on that context any further, you get this diagram. So that whole red bar is now this little red slice, and it's between the stars forming and all the stars burning out and either decaying or ripping apart. And in this context, maybe the conclusion you draw is, well, none of this matters, and we can actually cut the talk short and just head over to the bar, which you will surely see me at later tonight. But rather than pull back, what I'm going to do is pull in. So there are four things that I want you to take away by the end of the talk. So the first is that actually it's going to be really valuable to broaden the ecosystem and bring pharma in particular. The second is that aging biology is a platform to address multiple diseases. I think everyone in this room knows that. And that therefore pharma really should be super interested in getting in. But the commercial market is not there yet, number three. And so the last item is number four. There's actually a job in front of us, in front of the folks in this room, to build the case and bring others in. So I already made kind of a hand-wavy argument on the first one, going back to John Maynard Smith. But here's a little kind of more direct argument, which is looking at the data around what's driven or what's been involved, at least, with new molecular entities, that's drugs approved by the FDA over the last 30 years. And this is just a chart of the top 20 pharma. Obviously, it would be an even higher line if I did top 50. But you can see pharma is involved in the majority of medicines that make it to market. And most of the exceptions here are actually rare disease, which of course matter a lot. But if we think about the chronic diseases that drive the bulk of burden of disease on the world, this is an even higher chart. I'm not going to belabor the first point. That's really all I'm going to show, because I think folks understand the point of like bringing in other parties is going to be valuable. Even though, frankly, most of the, not shown on that chart, but most of the ideas start in academia and biotech, and it's only pharma that's helping with the translation part. So the second point I want to make is just how important aging biology is as a platform to address disease. I think kind of everyone in this room knows that, but unfortunately it's a message that still is not out there enough outside this room, and so we have a job to play in proselytizing to get that message out. And the logic goes like this. So the number one correlate of disease is a kind of famous chart that shows up in a lot of presentations. The number one correlate of many of these chronic diseases is age. It's the best predictor of whether you're in a disease state or not. And you can see that it's going up exponentially across the four shown here: cardiovascular disease, cancer, dementia, diabetes, which are really the scourges of humankind. We haven't thought of this as an intervention point historically, because we've thought of age as not something that you can manipulate. But if you can, in fact, decouple biological age from chronological age, it becomes a very powerful intervention point to potentially address multiple diseases. The question is there science that actually makes us believe that? And I'm in a room full of scientific specialists around this, but I'll have the temerity to actually build out the case a little bit. Which is there's actually a lot of data to say that lifespan is an evolved and genetically regulated trait, and that implies that it is malleable. The first line of evidence is just theoretical. Energy and resources are not infinite. There must be an optimum in terms of how those are deployed between growth and reproduction on the one hand and maintenance repair on the other. That optimum is set based on what's going to maximize the long-run rate of increase of a line of descent. It is not set based on what's going to maximize your own individual lifespan. And that implies that there is an optimum lifespan that will vary by each species condition or niche, as well as what's happening in the local environment. And that's exactly what we see. In comparative biology, closely related species in different niches can have vastly different lifespans. So with this colorful diagram on the top is, I'm sure, incredibly hard to read, but it's the radiation of Pacific rockfish that vary in lifespan from 10 years to 200 years based on size and depth, which correlate with predation risk and resource variability. We even see this within a species. So the bottom chart, you've got, you can think of queen and worker ant. In fact, they could even be clones. They have the exact same DNA. The only thing that's different is what's turned on and off epigenetically. In the common black ant, a worker ant lives 10 months. A queen lives 30 years. And what's shown in the diagram on the bottom is that that's a general feature of eusocial insects. And of course, at the very bottom there is just the note that organisms without a soma don't age, or those that can revert to germ state, like the immortal jellyfish. And of course, that's an aspect of biology that's under tremendously strong selection, right? That the germline would be immortal. Sort of, anything that doesn't reach that will go extinct. And that's now being, exploited, hopefully, with partial reprogramming. There's a third line of evidence around plasticity based on conditions. It's kind of a general feature that if you stress an organism in various ways, as long as it's not too great, it would actually live longer. Which seems incredibly counterintuitive until you think of it in light of the first column, which is it's an adaptive strategy to redeploy resources from growth and reproduction to maintenance and repair to wait for a breeding opportunity. It's likely, it's a little bit of a fitness reduction because you're delaying a breeding opportunity, but of course it's better than trying to breed when it's not possible. And what's shown here are data on the top. Longevity data. This is McKay's work back in 1934 in rodents. And on the bottom is health span data from University of Wisconsin. Those are two rhesus monkeys of the same age. The one on top has been fed ad lib, and the one on bottom has been caloric restricted. And you can kind of even visually see that there was a health span difference. That's also true in humans. So the top is epidemiological data from Phelan and Rose, looking at different populations based on caloric intake and lifespan. And the bottom is some healthiness data from a crony before and after caloric restriction. Of course, quite famously, we see it with genetic variants. Cynthia Kenyon's kind of very famous paper that kicked off the field, or at least accelerated the field, on DAF2 in worms. And the bottom data there is human centenarian data on the orthologous IGF-1R, showing that there's variants and that those variants have some effect. And then lastly, there's pharmacological interventions, right? The ITP from the NIA showed more than a decade ago now that you can extend the lifespan of a mammal with a pharmacological intervention with rapamycin and many others since then. And literally the only thing that's missing on this entire page is that last gray square, which is the proof that you can change health span and lifespan in humans. But the logic chain is very clear that that last piece of this page will eventually be filled in. So that's kind of a quick rundown of why this matters so much. And I didn't show it, but Linda mentioned it. It's not just lifespan in those data, right? You're affecting multiple disease states at the same time. So where is the industry with this? Unfortunately, not yet where it needs to be. It's starting, but it's not far enough to really drive a load of translation, which is what we want to see. So we're seeing the kind of starting green shoots. So the earliest lead indicator is research activity, which is largely driven by researchers. It's in the first column. That has been going up, and it's been going up faster than the kind of baseline of biomedical research, 12% per annum, publications in this field versus a baseline rate of 3%. Kind of the next more proximate leading indicator, which is what's happening with biotechs. You see the total funding and total number of companies here, and since 2016, you see the same thing. It's been outpacing. So there's been a 9-fold increase over that period in the funding relative to kind of a biotech baseline of only 5%. And of course, we also have some very high-profile examples like Calico and Altos that are super important because they capture the imagination and make people kind of pay attention to the area. The result of that is that there's now more than 150 companies with $9 billion in investment, and this is kind of a clustering that can emerge if you don't train, just ask it to cluster. Here's a different clustering. This is what's actually now in the clinic from that set of companies, and now it's organized by the hallmarks of aging on the vertical axis here, which, as Felipe pointed out, and even the original authors when they wrote the paper, look, it's an imperfect framework. We don't yet know what's causal and so on, but it's still probably the best organizing framework we have. And you can see that there's a small but growing pipeline that kind of cut across most of the hallmarks. Maybe the better view is this page, which is how those then cut across different therapeutic areas. And you can see for such a really small pipeline. I mean, a single pharma company could have easily a pipeline larger than what's on this entire industry page right now. For really small pipeline, it actually cuts across a large range of therapeutic areas, which is a kind of implicit validation of the thesis that aging biology hits a lot of different disease states. What's missing from this page, so if you look, I mean, it's probably too small to see, but what's missing from that page is any big pharma company that's actively directly involved. The best we can say right now is that there's some deal making happening, which of course is another kind of leading indicator to getting direct pipelines around this. And you see some multi-billion-dollar investments from AbbVie and Novartis, and you see others, Regeneron, Merck, and others that are making some initial plays in this space. So it's promising, but it's not far enough, which gets us to point number 4, which is we therefore have a job to play. And of course, it's going to come about in the natural course of your work, but there are some ways to think about this that I think can help direct where efforts are applied. So I'm going to tell you how I think about this. It's a little bit involved, so let me start with the big picture, which is ultimately it's a theory of constraint. There are a set of things that have to fall into place for a drug to get translated. All of those conditions need to be in place, and your effort should therefore be focused on the things that are the limiting factor and not overworking the things that which we've already resolved. So let me walk you through the hypothesis, and then I'll show you some data. So the framework for the hypothesis is that there's a set of things that need to come into place. You need to target the right biology. Per the last talk, you don't actually need to know that you're targeting the right biology, but so phenotypic screens actually work fine, but you actually do need to target something that is relevant biologically to the disease. You need some kind of chemistry that can modulate the target. I use chemistry in the very broad sense of the word. It doesn't have to be a chemical molecule that's directly interacting. Ideally, you have a model system in which you can optimize. That model system should be predictive of what happens in humans, and one that you can iterate on quickly. And then in the clinic, you need to be able to identify the right patients and measure their response with biomarkers. That's the framework. I'll bring it to life with an example, which is hepatitis C. So hepatitis C is a disease that's been effectively cured in our lifetimes, a huge accomplishment. If you use this framework and you looked at what was happening in the 90s, where there was a tremendous amount of effort, unfortunately a tremendous amount of effort that ended up proving fruitless, and that led to the demise of a number of companies that were really great companies like Chiron. If you had used this framework, you would have said, well, yes, many of the conditions are in place. We understand there's only a handful of viral targets. We can access them with small molecules, and we can easily measure patient response and identify them with a simple blood test and SVR. But critically, what was missing is there was no model system because you couldn't grow hep C in a petri dish, unlike a lot of other infectious diseases. The conclusion you would have said is, okay, we're stymied until we fix that barrier. In 1999, the replicon mouse was created, and using the framework, you said, okay, progress should now be rapid. And that's exactly what happened. Just a couple years later, Pharmaset ran the screens that led ultimately to Sovaldi. So that's an anecdote. That's N of one example, but you can look at this systematically, which is what we did. So you can think of a case control study. Cases are diseases in which we've made progress. Controls are diseases in which we've failed to make progress so far. We looked at across 16 therapeutic areas. We picked the 2 most prevalent indications in each category after some exceptions. And then we had a set of physicians blindedly, so we didn't explain what we were doing, but we asked them to score this whole long list of disease areas across a range of criteria that matched the categories that I was describing, and fairly objective kind of rules in which they applied. And the results largely validate the thesis that yes, these conditions matter. Not shown here, actually, the interaction effect also matters. So it's not just about them independently happening, but it is the multiple happening that it is important. And so when you think about making progress in the disease that you're working on, obviously this is going to vary by the biology you're working on and the indication. But the way to think about it is make sure you're thinking through all the constraints and you're spending enough attention on the one that is the lacking one. There are probably some general features that I can describe here. So when we looked at what's the last constraint that falls usually, actually half the time the hepatitis C story is actually very illustrative, because half the time the last thing that falls is the model system. Another one that's often a problem is the biomarker. And so obviously this is going to vary by situation, but there's some general themes to think about where, as a group, we might be under-investing versus over-investing. So are we really thinking about the model systems enough? It's a lot of pharma doesn't pay too much attention to creating new model systems, and a lot of research is actually more on the pathways, right? There's tremendous attention from the VCs on modalities, but there may be a piece that we're leaving out a little bit. Okay, so I know there's a bit of a whirlwind, starting with the Earth forming, but tried to get through these 4 points and leave enough time for questions. So the first is, there is value in building this ecosystem, continuing to broaden the types of participants, as well as the number within each group, that we have this amazing platform that should address multiple diseases, sort of the best untapped vein I can think of for new therapies. Pharma should therefore be interested, but they're not quite there yet. There are some things we can do to get to proof of concept that will then, I think, start to bring in these other parties. So happy to take questions, accusations, whatever kind of commentary is helpful.
Thank you so much. Thank you so much, Michael. Fantastic presentation. Very good to put it in perspective. Alex has a question.
Hi, Michael. Actually, apologies for missing the beginning of the talk. I was outside this hall with Big Pharma talking about aging. So first of all, huge thank you for coming and presenting those slides. I know that some companies pay hundreds of millions to get to those slides, digest the strategy, and then make multi-billion-dollar decisions. And of course, also by the way of kind of additional introduction, Michael Ringel is probably the most informed person in the pharmaceutical industry in general, but also in longevity. So thank you for your efforts. I have two very quick questions. So one is, many pharmaceutical industries are interested in this area. Many of them are trying. What is the best way for a pharmaceutical company to get into aging and to make a really big bet? What do you think is the best strategy, aside from hiring BCG, of course? And the second question is that for a small biotech that is at this conference, and by the way, there are some pharma companies here too, but for small biotechs, how they should formulate the strategies at this point in time in order to have a successful partnership with a big pharma company?
Yeah, great question. So on the first one, imagine you're a pharma company and you're working on a disease state that has been tough to address with existing approaches. I mean, there's a long list of these. You could say Alzheimer's with A-beta would be an example. Hopefully, we'll still make progress using that approach. But my whole career, we've been working on A-beta. And there are some other orthogonal approaches, right, that come from aging biology that might be helpful. Pharma often waits to see proof of concept around an area before moving in, and then valuations shoot way up, right? Now, I think as you described yesterday, right, there's a sort of risk and payout that you want to think about, right, which is, well, obviously once you wait for proof of concept, things have been de-risked, and you should be willing to pay more. But there tends to be a phenomenon of relatively small number of sellers and a lot of buyers when that happens. And so if I were advising a pharma company, I would say think about getting in now ahead of what I think is kind of a relatively low-risk bet that aging biology is going to pay off. And once POC happens, right, there's going to be a swarm. So act now before the swarm hits. That would be advice to them. On your second question, advice to biotech. I mean, there is no substitute for POC in being able to make the case. POC being proof of concept, right? The mechanism is in fact working in humans, and so find the critical path that gets you there. And again, sometimes it's not the most obvious thing on that list of constraints that I was trying to raise.
Thank you. Other questions?
I think we have to...
Maybe one.
A very quick one. Then.
Okay, I'll keep it a short answer then.
There was
Anybody?
someone here, I think. I don't know.
I've got a mic, so anybody wants to ask a question?
There's there in the back.
Okay.
Rafa.
A quick one. Very quick one.
So, from what I understand, AbbVie, like they involved with Calico, but when you speak to people involved in neurodegeneration in AbbVie, they totally separate from the AbbVie Calico aging thing. So how do you get people from AbbVie neurodegeneration to say, well, actually, maybe we should talk to the AbbVie aging Calico collaboration?
So I'm not going to comment on any particular company,
No.
but
For
sort of
example,
the general question of how do you get biologists in a pharma company interested in talking to a biotech. Look, I mean, I think this is where outreach from the biotech can be helpful. That can be through, obviously, BD groups within pharma, but also scientist-to-scientist outreach I would totally encourage. We've got people who've crossed labs and things like that. And then, of course, pharma is actively reaching out, and it's part of the job of people in the business development group, right, or partnering group, whatever it is. So hopefully there's also inbounds coming to the small biotechs, and obviously be ready for those.
All right, thank you so much. Let's give Michael another round of applause. And we have a break now